Hi, my name is Kayla Henry, and my project is on the synthesis of photophysical properties of lanthanide X2 phenanthroline N PTA 7-N-NO3 towards water-soluble luminescent lanthanide complexes with bactericidal properties. An important chemical property of 1,10-phenanthroline is that it is a chelating agent, and this is just meaning that it can bind tightly to metal ions. It is important currently in undergoing medical research as 1,10-phenanthroline is being used to bind to toxic materials in the body. In addition, 1,10-phenanthroline also produces color in complexes during the presence of metal to ligand or ligand to metal charge transfer electronic transitions. This is just saying that the electron on the center of the metal can be transferred to that phenanthroline ligand or vice versa for the ligand to metal charge transfer. In addition, there have been some interesting shapes and structures that have been seen in previous research with 1,10-phenanthroline. But while I have been looking into previous research, I did notice that there were fewer studies on the phenanthroline's isomer 1,7 and 4,7, and this is presumably due to the distal nature of that second nitrogen, as it can make it very difficult for cleation to occur. This study is reporting the attempted synthesis of lanthanide complexes containing 1,10, 4,7, and even sometimes 1,7 phenanthroline, and also the compound 1,3,5 triazosebent phosphoadamintane, also known as PTA. PTA is important as it is used as a water-soluble component in some of the reactions to try and help bind the compounds. In addition, the lanthanides used for this project are europium, terbium, and holmium because they have been seen in previous research to exhibit luminescent components. So this is just describing the structure of some of the compounds used within the study. Figure 1 and figure 2 are both phenanthrolines, figure 1 being 1,7 and figure 2 being 4,7. And as you can see, these nitrogens are quite far apart from one another, and making that cleation, as I discussed earlier, quite difficult compared to 1,10, which are right next to each other. In addition, I did include the structure of PTA as just kind of getting an idea of the physical component of this water-soluble compound. So the experimental design and procedure of this study is relatively simple. So I chose to do 3 reactions. The first is including all 3 components, the lanthanide nitrate, the phenanthroline, and the PTA. The second reaction is just including the PTA and the lanthanide nitrate, while the third is getting rid of that PTA and instead just using phenanthroline and the lanthanide nitrate. And the process is relatively simple, in which the amount of grams is measured depending on the reaction and the reactants. And then those powders are combined into a round bottom flask with 50 milliliters of ethanol. The solution is then stirred via a magnetic stirring rod for 24 hours. After that 24 hours has elapsed, it's poured into an evaporating dish so all that ethanol can evaporate off. And once that happens, the powder is scraped from the evaporating dish and washed with ether. I'm currently in the stage of crystallizing these products using solvent diffusion. The process of solvent diffusion is using 2 vials, one inside the other, in which the outside vial has a more volatile solvent and the inside less volatile. The outside I'm using as ether and the inside is using as ethanol. In addition, in the ethanol, I will have a little bit of the collected product. The process is relatively easy, in which the volatile solvent will diffuse into the inner smaller vial and lowering that solubility of the product to eventually produce crystals. And this figure shown on the slide is an example of the first reaction using terbium nitrate and 1,10-phenanthroline along with PTA. So this is just showing some of the results that I have achieved within the study. The first one is using that second reaction with just europium nitrate and PTA. And then as you can see, it kind of has like a reddish pink complex. The second is that figure from the previous slide, just in powdered form of the first reaction with terbium nitrate, PTA, and 1,10-phenanthroline. The third one is I actually did a competition between 4,7-phenanthroline and 1,7-phenanthroline because I wanted to see which one bound better to europium nitrate. And I discovered that I felt that 4,7-phenanthroline bound better. And as you can see, its product is in figure 4 with europium nitrate and 4,7-phenanthroline showing a kind of yellowish complex. So while this study is in its initial stages, as you can see, results have been collected. Those bright pink and green luminescent colors seen on the previous slide are characteristics of F to F transition orbitals, which is just saying that electrons are being shared between the F orbitals. An important aspect of this is that it can be photosynthesized by ligand absorption bands using the FRET mechanism to try and get a better idea of what's going on exactly with those electrons. In addition, the orange emission or that yellowish emission seen in figure 4 is either due to the ligand to metal excited state or the metal to ligand excited state. And another important chemical aspect of this project is that it can help create a structure for designing luminescent phenanthrolene lanthanide compounds. And while there is a chemical component, there is also a biological component, as the phenanthrolines have been known to insert into the minor groove of DNA. And this is done by the excited electron from that metal to ligand state is actually inserting into the DNA and cleaving the DNA. And this exhibits bactericidal properties. This is really interesting and undergoing in current research projects as it's being used to contribute to cancer and tumor targeted studies. And I'm hoping as I continue and further my work that my work on 4,7-phenanthroline will be able to use as a potential application to anti-cancer agents. This is just a slide acknowledging the references used in the previous slide for future works and conclusions. And lastly, this is my overall project again, or poster again. I just want to take a moment to really thank you for listening to what I've been doing within my research. And I want to take a moment to also thank Dr. Casadonte and the Biochemistry and Chemistry department for encouraging and promoting my research. And I do hope that you have a great rest of your time with URC. And thank you very much for your time.